Compare and contrast an ion channel receptor and a G protein coupled receptor. So in both of these examples, an ion channel receptor as well as a G protein receptor are going to be involved with converting some type of external signal into an internal response by the cell. The first one we're going to take a look at is going to be an ion channel, more accurately referred to as a ligand-gated ion channel. And so we've got an ion channel over here. In this case, on the right-hand side, we've got the closed position. And so we've got a transmembrane protein that is going to form a hydrophilic pore through the membrane and so allow for the passage of ions, small charged particles, to be able to pass through this hydrophilic pore as opposed to be blocked by the lipid tails within our phospholipid bilayer, that hydrophobic environment here. So what is going to happen is our external signal, our messenger, and so you can think about this as a neurotransmitter or in some cases a hormone, but what's going to happen is that messenger is going to act as a ligand. It's going to bind to our receptor, and as an action of binding to that receptor, we're going to see a conformational or shape change in our ion channel. We're basically going to change the protein configuration, and we're going to open up that pore. So in the closed position here, it's blocking the flow of ions. In that open position, ions are going to be able to flow down their electrochemical gradient, and so flow from high concentration to low concentration, and then carry their charges with them. And because they are charged particles, the charge inside and outside the cell could have an effect as well. So in this case, what we're looking at with this receptor coupled to an ion channel is that binding to that receptor is going to open up the ion channel. It's going to allow for ions to flow across the membrane, and in doing so, that change in the permeability of the membrane to those ions is going to change the charge differential, what's going to be referred to as the potential of the membrane, but it's going to basically change the charges in relationship to outside and inside of that membrane. This is going to be important when we take a look at nerve cells and muscle cells, because that ion flow, that change in the electrical potential of the membrane, is going to be important for sending nervous system signals as well as coordinating the contraction of a muscle cell. The second example of a receptor coupled to a mechanism to convert the signal, transduce the signal from an external signal to an internal signal, is taking a look at G protein coupled receptors. And there are actually two forms of G protein coupled receptors, and we'll look at each one individually. Now, the first one to take a look at is a G protein coupled receptor that will ultimately interact with adenyl cyclase and result in the production of cyclic AMP. Now, the same basic idea is going to be present that we saw with the ion channel. We're going to have an external signal outside that we need to convert into some type of internal signal. Now, in the case of a G protein coupled receptor, we're not looking at an ion channel. We're looking at a transmembrane protein that is going to interact with another protein along the cytoplasmic side of the membrane. And that other protein is going to be a G protein, which basically comes along with the G protein coupled receptor. When the appropriate ligand binds to that, the appropriate signaling molecule or messenger molecule binds to our receptor here, our G protein coupled receptor, this is now going to activate our G protein. And that G protein then is going to interact with adenyl cyclase. This is going to be an enzyme that is embedded within the membrane. And what's going to happen when we activate adenyl cyclase is we are going to convert ATP into cyclic AMP. And this cyclic AMP is going to be referred to as a second messenger. The first messenger is the ligand, the hormone, the messenger outside of the cell. The second messenger is going to be the message inside of the cell. And so cyclic AMP as a second messenger is going to act inside of the cytoplasm. And normally this is going to be involved with activating a series of protein kinases. Now, a kinase is going to be an enzyme that involves transferring a phosphate group from ATP onto a protein. And so basically a protein kinase is going to be involved with energizing proteins within the cell and basically making them active. And so what we're going to have is going to be a phosphorylated protein. You can think about this as being an activated protein. A protein is going to be capable of doing what it needs to do within the cell. And so in that way, this phosphorylated protein is then going to trigger a cell reaction. It's going to control some type of process within the cell, but it is basically going to trigger what is going to be that cellular response. What's important to recognize with this G protein coupled receptor with the adenyl cyclase and the cyclic AMP is that we can amplify that external signal by producing multiple copies of our cyclic AMP down here. And so we got one signaling molecule outside of the cell, resulting in multiple cyclic AMPs acting on multiple protein kinases, resulting in multiple phosphorylated proteins and a much more robust, stronger cell response within the cytoplasm of this cell. The second example that we're going to take a look at in terms of G protein coupled receptors, in this case, we're going to have the G protein receptor, which is going to be coupled to basically is going to activate our phospholipase C, and that phospholipase C is then going to result in the production of DAG and IP3, both of which are difficult to spell and even harder to pronounce. And so the same basic idea is going to be present. We're going to have a transmembrane protein that is going to convert that external signal, and so a hormone or a ligand or a messenger is going to bind to this transmembrane protein, serving as a receptor. That is then going to activate a G protein, but in this case, that G protein then is going to act on phospholipase C, another protein that is embedded within the membrane. That phospholipase C is going to do a couple things. It can involve the production of DAG, and DAG can activate another set of protein kinases. And so DAG is going to affect protein activation by activating the protein kinases, resulting in phosphorylated proteins and allowing those proteins to have a response within the cell. It's also going to be involved, the phospholipase C is also going to be involved in the production of IP3. The IP3, again, is another example of a second messenger. So in this case, both DAG and IP3 are second messengers. IP3 is going to act on other regions of the cell, and as it's illustrated here, it is going to act on a ion channel and regulate the opening of a calcium channel, and that release of calcium then could have an effect of controlling different things within the cell. And so with these G protein coupled receptors, we're still taking that external message outside, that external hormone or ligand. We are converting it into a second messenger that is going to be acting inside of the cell to trigger a response of the cell. And protein activity in the response in terms of the protein kinase activated by DAG, or this influx of calcium that will control other things within the cell.